Hi everyone. Today we will present the structural analysis of our chiral compound 16, and is a PAR inhibitor. We are group number 1. Our chiral compound is talazoparib. Is an FDA-approved drug for treating BRCA mutated breast cancer and HRR mutated prostate cancer. Talazoparib has two pharmacological targets, PAR1 and PAR2. PAR enzymes in general are essential for repairing DNA damage of single-strand breaks that enable DNA replication and tumor survival. With the inhibition of these enzymes, it will increase the cell apoptosis. PAR1 has larger and more complex protein structure comparing to PAR2, with smaller and less complex structure consisting of primary DNA binding and catalytic domains. PAR2 catalytic activity is depending on the NAD+. Talazoparib has binding affinity for both targets, but is more potent at binding PAR2 with IC50 of 0.2 comparing to PAR1 with IC50 of 0.6. It also exhibit higher binding affinity for PAR2 compared to PAR1. In that case, for this project, we will use PAR2 as a target for investigating and analyzing our protein structure in complex with our compound. The aim of our project is to identify the putative of target, predict the epitopes for target protein, and investigate the compact of genetic variation and species-specific effects. And we are using Chimera X for analysis. And for more in detail, you can find it from this poster.
The PARP2 protein in figure 1c contains non-standard residues, talazoparib and glycerol. As shown in figure 2c, key and AD+ binding left residues include leucine 429, serine 470, and tyrosine 473, with a catalytic residue being glutamate 558. Potential immune recognition sites on the PARP2 protein were identified by predicting linear and conformational epitopes. The 3 longest linear and 2 longest conformational epitopes were identified from the protein structure and colored with different colors in figure 1b.
And then to our docking results. So you can see in figure 2e that talazoparib docked to the mouse model and that talazoparib clashes with the catalytic residue glutamic acids and forms hydrogen bonds to glycine and serine. And you can see in figure 2d talazoparib docked to the human PARP2 and that it forms hydrogen bonds with glycine. And based on our analysis, so if you compare the docking results to analysis of talazoparib bound to PARP2 in figure 2c, you can see that there are differences that are at least partially probably due to challenges with the docking. And the sequence identity between human and mouse model was 88%. Despite this, the mouse PARP2 catalytic domain structure is very well conserved. So when comparing heavy atom positions, it gives us a root mean squared deviation value of only 0.8 angstrom. And because of this low RMSD value, mouse PARP2 is a good model for in silico analysis. In vivo, PARP2 interacts with histone pyrrolation factor 1 and other proteins, and due to this structural analysis of one protein in the base excision repair pathway is not enough on its own to determine if mouse is a good model organism in vivo. And then to our mutant homology model. We mutated arginine in position 570 to proline. And as you can see in figure 2b, arginine forms 3 hydrogen bonds and is part of the beta sheet. But if you look at figure 2a, you can see that proline does not form any hydrogen bonds, and in this mutant, the beta-14 strand is impacted. And this disruption of hydrogen bonds is likely to impact the catalytic domain, despite the fact that the mutation is located 21 angstrom from the catalytic cleft. And when we looked at mutations in the 570 position, so we found either arginine mutated to glutamine or tryptophan in actual cellular samples. So we found 5 cancer tissue samples with these mutations in the catalog of somatic mutations in cancer database.
For the putative off target, we were identified that primarily there are the enzymes and prediction from the polypharmacology Browers 2 and sweet target prediction show really low reliability. In contrast with the SIA analysis suggests strong off-target potential with a p-value less than 0.5 for PAR16, PAR4, and TNKS2. And additionally, the superpret identify high off-target probability, more than 95% for HIF1 alpha and NF kappa B1. That's why for further investigation of this possible putative off-target is needed to assess the possible unintended pharmacological effects. Thank you.